Greetings, everyone. I'm Leandro Mfutumukiza, a student from Ines Ruhengeri, Faculty of Health Sciences, Department Biomedical Laboratory Sciences in Level 3. And I'm going to do some presentations in this module, Applied Immunology. And I will present about cell therapy, answering these two questions, which are what is cell therapy and how does it work, and what are the risks of cell therapy. And again, I will present about immune interaction with the vaccines, with its two questions, stating how do adjuvants improve immune interaction with the vaccines, and what is the role of memory cells in vaccine efficacy. As I have said, at first we have cell therapy. And its first question is about what is cell therapy and how does it work? And cell therapy refers to the use of antiserum, which is blood serum that contains high concentrations of antibodies or a blood-derived product containing antibodies to treat or prevent various infections or other diseases. The term cell therapy is most commonly associated with the use of immunoglobulin preparations containing antibodies from human or animal sources to provide passive immunity against a specific pathogen or toxin. It is a form of passive immunization where the individual receives preformed antibodies rather than having to produce their own through an active immune response. And this cell therapy works through different ways, which are at first antibody transfer. In this antibody transfer, it is the primary mechanism of cell therapy, where the antibody transferred are from a donor, usually an animal like a horse or in some cases humans, to the patient. And these antibodies have been produced in response to exposure to a specific pathogen, such as bacteria, virus, or toxin. Then there is neutralization of pathogens in cases of viral infections or bacterial infections. This neutralization of pathogens, once administered, the antibodies in the serum recognize and bind to the target pathogen or toxin. This binding can neutralize the pathogen directly, preventing it from infecting cells. For example, in cases of viral infections, as I said, antibodies can prevent the virus from entering host cells. Even in bacterial infections, antibodies can block the bacteria from attaching to host cells or mark the bacteria for destruction by other immune cells in a process known as opsonization. Then there is complement activation, where many of the antibodies used in cell therapy can also activate the complement system, which is a part of the immune system that helps to kill pathogens by promoting inflammation. Even this complement system helping promoting phagocytosis and the destruction of pathogens through the formation of a membrane attack complex. Then there is toxin neutralization. In cases where cell therapy is used for antitoxin therapy, example for diseases caused by bacterial toxins like diphtheria or tetanus, the antibodies neutralize the harmful effects of the toxin rather than targeting the pathogen itself. For instance, tetanus antitoxin can neutralize the tetanus toxin produced by Clostridium tetany before it causes severe neurological damage. Again, there is a rapid action because serotherapy involves the direct administration of preformed antibodies. It provides immediate protection or treatment, and this is specifically used in stimulation where a rapid immune response is needed. Then there is the second question about this serotherapy is stating or directing us to the risks of serotherapy. And those risks are, at first, there is, or there are allergic reactions. In these allergic reactions, we found hypersensitivity, where the patient may develop an allergic reaction to the foreign proteins in the serum, especially if it's derived from animals. And these reactions can range from mild rash and fever to more severe anaphylaxis, which is a life-threatening whole body allergic response. Another thing that can be found in these allergic reactions is the serum sickness. A delayed hypersensitivity reaction called the serum sickness can occur days to weeks after receiving animal-derived antiserum, and symptoms include fever, joint pain, rash, and swelling. And this is an immune response to the foreign proteins in the serum, particularly when animal-derived antibodies are used. Then there is a transmission of infectious agents, where these infections may be from contaminated serum. In rare cases, where antiserum derived from animal sources may carry infectious agents, especially if the serum have not been thoroughly screened or treated. Again, there may be viral transmission as a potential risk of transmitting viruses such as hepatitis or HIV if the antiserum comes from human donors who were not adequately screened. And the next risk, the other risk is immune complex formation, where by the introduction of antibodies from an external source can sometimes lead to the formation of immune complexes in the body. And these complexes, as antibody-antigen aggregates, can deposit in tissues such as the kidneys or joints, leading to inflammation and damage, a condition known as immune complex disease. Then there is immunogenicity, which is an immune response against the antiserum. In some cases, the recipient immune system may recognize the foreign antibodies as invader or mount an immune response against them, reducing the efficacy of the treatment. Then there is a limited duration of action. By this, you have temporary protection, where serotherapy provides passive immunity, meaning that the protection lasts only as long as the administered antibodies are present in the body, usually weeks to months. After this period, the person no longer has protection from the pathogen or toxin, and further treatment or vaccination may be needed. Then there is overdose or incorrect dosing, which is an excessive antibody dosage. If too much antiserum is given, there is a risk of excessive immune system activation, which could cause systemic inflammation, organ damage, or shock. Then proper dosing is crucial for minimizing side effects. At last, there is a lack of long-term immunity. Since serotherapy provides passive immunity, it does not stimulate the patient's own immune system to produce a long-lasting response. Therefore, the individual remains vulnerable to future infections with the same pathogen unless active immunization is given later, which is vaccine. Then after this, Let's talk about immune interaction with the vaccines. And the first question about this topic is how do adjuvants improve immune interactions with the vaccines? And these adjuvants are substances that are added to vaccines or immunotherapies to enhance and trigger the body's immune response to an antigen. They do improve immune interactions with the vaccines by enhancing through various mechanisms that stimulate the immune system, prolong the immune response, and enhance the immune recognition of the vaccine's antigen. And here's how adjuvants enhance the immune response. At first, there is activation of innate immune cells, and by this, adjuvants primarily stimulate the innate immune system, which is the first line of defense, by activating innate immune cells. And the adjuvants help trigger the adaptive immune response, which is more specific and long lasting and more quickly and even robustly. And by this, it is by the help of pattern recognition, where many adjuvants activate pattern recognition receptors, such as toll-like receptors, which recognize pathogen-associated molecular patterns on immune cells such as dendritic cells, macrophages, and neutrophils. And again, it is by the help of cytokinase release. Upon activation, immune cells release cytokines, which are interleukins and interferons, and they are chemical signals that coordinate the immune response, and they help to activate other immune cells, recruit them to the site of the vaccine injection, and create a pro-inflammatory environment that enhances the immune response. Again, we have enhanced antigen presentation to ensure the vaccine's antigen as efficiently recognized by T cells. Adjuvants improve the ability of antigen presenting cells, such as dendritic cells, to capture, process, and present the vaccine antigen to T cells. And this is done by improved uptake of antigens, where some adjuvants increase the ability of antigen presenting cells to take up and process the vaccine antigen. Again, by the activation of T cells, where once antigen presenting cells process the antigen, they present small fragments or peptides of the antigen on major histocompatibility complex molecules on their surface. This allows them to activate CD4 helper T cells, which promote B cells activation, antibody production, and CD8 cytotoxic T cells, which recognize and kill infected cells. Then there is prolonged exposure to the antigen, giving the immune system more time to react. At fourth, there is activation of the adaptive immune system or increased immune cell activation for stronger and longer lasting immunity. This by the adjuvants indirectly boost the adaptive immune response, the more specific immune system that generates long-term immunity by enhancing the response of B cells and T cells. And this B cell activation, in addition to activating T cells, adjuvants can stimulate B cells, which are responsible for producing antibodies that neutralize pathogens. And memory immune cells, adjuvants can also help stimulate the formation of memory B cells and memory T cells, which remember the pathogen and provide long-term immunity. Then at fifth, there is a modulation of immune response quality. And this depends on the pathogen, where some adjuvants can influence the type of immune response generated, helping tailor the vaccine to be more effective against particular types of pathogens. And this by Th1 and Th2 responses, where adjuvants can skew the immune response toward Th1 or cell-mediated or Th2, which is humoral antibody-mediated response, depending on the type of pathogen the vaccine is designed to protect against. For instance, Th1 responses are more effective against viral infection, while Th2 responses are better for defending against extracellular pathogens like bacteria. And again, there is in this modulation of immune response quality, there is enhancement of cross-protective immunity, where certain adjuvants can also help generate broader protection against related strains or variants of a pathogen by inducing cross-reactive immune responses. Then there is to the last question, what is the role of memory cells in vaccine efficacy? The primary goal for a vaccine is to mimic a natural infection without causing disease, thereby stimulating the immune system to generate memory cells. That's the primary goal of vaccine. And effective vaccines generate a strong and durable memory cells or durable memory cell response. For instance, vaccines like the measles, mumps, rubella vaccine or HPV vaccine stimulate long-lasting memory B and T cells, which provide protection even after the antibody levels decline. And memory B cells provide long-lasting antibody protection by rapidly producing high-affinity antibodies when re-exposed to a pathogen. And memory T cells provide faster, more effective cellular responses, helping to eliminate infected cells and coordinate other parts of the immune system. Memory cells ensure that the body is ready to respond quickly and robustly upon encountering a pathogen it has previously been exposed to, whether through natural infection or vaccination. And then the presence of memory cells is the foundation of long-term immunity, allowing vaccines to provide lasting protection, sometimes for a lifetime, against certain diseases. Thank you very much for your kind attention, and I hope this will be helpful